Okay, now we will discuss about the characterization of cell banks. That includes identity, stability, sterility, viability, growth characteristics, and so on. First of all, for identity or for identification in cell banking, we can use karyoology or chromosome analysis, isozyme analysis. In case of human cells, you can use DNA profiling. Human leukocyte antigens typing you can perform, and cell lines for are proteins that vector integrity, recombinant cell expression, plasmid copy number, insertions, deletions, you can all identify them easily. Next is the stability, which is very important for the cell banking. Once every 5 years for cryopreserved, genetic stability is very important, and global or target gene expression, proteomic or metabolic profiling can easily inform you or tell you about the stability. Phenotypic markers, viability, copy numbers of constructions, mRNA, protein levels, these all can be used as marker for the analysis of stability of cell banks. Viability test you can perform using the membrane integrity, metabolic activity, and DNA replication. If you talk about the growth characteristics of cell banks, then well understood that viability, morphology, cell doubling time, plating efficiency, these all basically associated with the growth characteristics in the cell bank. Tumorigenicity in case of CCLs or continuous cell lines, some contains oncogenes or virus to be immortal cells and classified as tumorigenic. Different assays can be used for analysis of tumorigenicity. In case of in vivo, you can inject cells into the immunocompromised animals, and CCLs or new DCLs, nodule growing and metastasis, you can check, and you can analyze the tumorigenicity. For oncogenicity could be checked in CCLs or SCLs. Animal models you can use newborn nude mice, newborn hamsters, and newborn rats could be used as animal models to check the oncogenicity. Cytogenic genetic test includes further DCL, SCL, or SCCLs. In case of DCLs, count of stained chromosomes, and in case of CCLs, marker chromosomes and support genetic stability. These can be checked. Testing of for adventitious agents. In vitro test you can use, or in vitro test for viruses you can use, such as cell culture safety test, transmission electron microscopy can be used, or biochemical tests. In case of in vitro test for non-viral agents, you can use the mycoplasma or bacterial and fungal sterility and mycobacterial testing. Characterization of seed bank. For the characterization of seed bank, you first of all you have to check the phenotype or morphology. For that you can use the microscope, that includes the bright field microscope or transmission electron microscope. While in case of genotyping, you can use DNA sequencing, recombinant proteins, and you can also use for the characterization of seed banks biochemical properties and immunological assays. Biological raw materials, that includes adventitious agents such as serum, trypsins, animals, acids, and other biological reagents. Cryocell International is the world's first private code blood bank. In 1992, the company was the first in the world to separate and preserve stem cells. Here is the link in which you can Google, or you can go get more information about the Cryocell International. There are 589 culture collections in 68 countries registered in the World Data Center for Microorganisms and World Federation for Culture Collections. Cell and tissue banks, internationally there is American type cell culture collections, Carolina Biological Supply Company, Here are some different agencies worldwide which are basically involved in cell and tissue banking. In case of microbial bank culture, the Wellcome bacterial collections, E. coli genetic stores and stock centers, and my mollictus collections, these are other microbial culture banks. In case of fungal culture cell banks, you can use the fungal genetic stock centers, University of Minnesota rhizobium collection and rhizobium cell culture collections, etc. Cell banks for algae are also present. Here is the list of cell banks which are basically involved in the preservation and collections of samples related to algae. Plant culture banks are also present. These are the some list which are associated with the plant culture saving or animal banks. Animal banks, cell banks includes the mouse stem cell banks, marine mammal tissue bank, ocean genome legacy, and here is also some other list of banks which are associated with the animal cell bank. So here are some cell and tissue banks in Pakistan. Cryocell Pakistan is its head office is in Karachi, and another branch is in Lahore. Thank you.